In my lab, we do primarily we do a mix of experimental evolution and theoretical population genetics. And the experiments, what we do is we take hundreds or sometimes thousands of populations of budding yeast, and we allow them to evolve in various kinds of laboratory conditions for thousands of generations. And then we try to characterize what happens over the course of this evolution by sequencing individuals or populations through time, by doing various phenotypic characterizations of the changes that occur during this evolution. And when I started doing these experiments a dozen years or so ago, I certainly had the impression, and I think most people in the field had the impression, that evolution in these kinds of systems was going to be, unless you tried hard to make it not, was typically going to be limited by mutations. So basically the primary thing you had to do in order for the population to evolve was you had to wait for the relevant mutations to occur, and then natural selection would act on those mutations, and you would see sort of sequences of selective sweeps, for example, as these populations evolved. And as we started to do these experiments and characterize them over the, this sort of has this ringing noise, and characterize them over the past 10 years or so, we've come to realize that this is actually a very atypical case. And much more commonly what seems to happen is that evolution is not mutation limited at all, and that rather mutations are occurring in a variety of genetic backgrounds within the population all at the same time. There's lots and lots of different loci at which mutations are occurring, and natural selection is acting on them all at once. And so this has got me interested in understanding what we expect to see theoretically in terms of how evolution proceeds and what patterns of genetic diversity we expect to see in populations in these kinds of situations where selection is pervasive. And I think over the past 10 years or so, it's become increasingly clear that this is a very common situation. So Richard, a couple of days, talked about situations where similar situations in influenza and HIV populations and people in human and Drosophila population genetics have also begun to see signatures of these effects. Is there a sort of ringing noise here? Anyone? No, it's just in my ears. Okay. All right. So usually I talk in these kinds of settings about the experimental data, but today I'm going to take the opportunity to talk about some of the theoretical work. And this is work that's about a year or so old, so I'm going to have to summarize that background, and then towards the end maybe I'll give a little bit of a hint of the future directions that we're going to go in. And so all of this work that I'm going to talk about is done primarily by a graduate student, Ben Good, in my lab. It's also done in collaboration with Richard and with Alexandra Walczek. And basically the question we want to ask is, if we're in a setting that's common in natural populations where the only information that we have access to is sequences of a few extant individuals from the population, what is it that we can infer about how selection pressures have acted? And actually, I'll address a simpler question, which is just when we have selection acting on many loci across the genome, what do we expect to see in this kind of data? So here I have an example from one of our yeast populations where we took 24 clones and we sequenced them, and I'm depicting the diversity that we observed in the sequence data by drawing the genealogy relating these clones inferred from that sequence data, the various relationships here, and here's a similar example of data from one piece of one of the influenza trees that Richard was talking about a couple of days ago. And particularly in these experiments, we have a tremendous amount more power due to the experimental setup than is present just in this kind of data. So in these populations, we know that there's selection acting on many of these mutations at the same time. But this is the kind of data we might be limited to in a natural population. And so what we want to understand is how does this pervasive selection shape these trees? How does it lead, what kinds of skewed branching or multiple merger or other sorts of non-neutral effects would we expect to see as a function of what types of selection pressures are acting? So that's the question we're going to address. Before I get into any detail, just 2 minutes, you all know this already, but just to emphasize the background, normally when we think about neutral coalescent theory, what we can do is we we characterize diversity by, first of all, thinking about the probability of genealogies relating a sample of individuals from some population. And then we think about separately calculating the probability of an observed diversity by imagining mutations occurring sort of at random on this genealogy. And that gives you some pattern that you see in sequencing individuals from within a population. And the two sort of key things, which I'm going to talk a lot about today, are the overall levels of diversity. And in the neutral case, we just have the pairwise diversity between, the average number of differences between two randomly sampled sequences is going to be proportional to the population size, of course, because probability of common ancestry is always going to be like 1 over n in a given generation. So the time scales are going to be of order n, where n is the population size, and thus the diversity, pairwise diversity, is going to be proportional to that. And the frequency spectrum in this neutral case is going to be like 1 over I. So basically the relative proportion of polymorphisms present in I out of n sampled individuals will go like 1 over I. And you all presumably know this already. Now the key difficulty, of course, you can obviously, as many of you have talked about, look at various complications to this simple neutral coalescent for various kinds of things like demography or spatial structure or the like. But the main difficulty when we think about selection is that the probability of the genealogy and the probability of the observed mutations given the genealogy are no longer independent things. Okay? And so, of course, there have been various approaches like the ancestral selection graph, for example, which give an in principle complete solution to this problem, but which are, in practice often computationally intractable. So what I'm going to focus on today is a sort of simple ad hoc approach where the goal is to allow us to make efficient computations of the expected patterns of diversity that we see when selection is common. Okay, so I'm going to get into the details of some specific model in a few minutes, but before I do that, I just want to sort of give the intuitive outline of the basic idea behind all of this work. So the basic idea is whenever you have a situation where selection is pervasive, you're going to have some variation in fitness within the population, and that's going to have some distribution, which I'm just drawing schematically here. And Richard talked about this. That's just a basic consequence of having selection acting on many loci at the same time. And if we sample individuals from within the population, they'll come at random from within this fitness distribution. Okay? And so if we think about the ancestry of these individuals, if we look backwards in time, okay, the ancestors of the individuals that we sample in the present will be from a more fit than average subset of the ancestral population. Right? And the reason for this is pretty straightforward. I like to just think about this in terms of of everybody in the room. It's sort of simple, intuitive thing to think about, right? Some of you guys in the room will presumably never have children. And so you are from this low end of the fitness distribution. This is you right here. But all of your parents have children, right? So your parents, on average, are more fit than you. And the further back in time we go, of course, the more true this becomes. Right? Farther back we go in time, the more fit subset of the ancestral population, you'll find the ancestors of the individuals present today. And of course, I mean, that's obvious if you think about it. That's what fitness means. The fittest individuals in the past are precisely those individuals likely to give rise to lots of offspring in the present. Right? And so, roughly speaking, what selection is going to do is lead to some kind of what will look like a reduction in the population size as we go backwards in time. Okay? And as we'll see, it also leads to other sorts of effects, particularly when selection is acting on lots of loci at once, like multiple merger events and a loss of exchangeability between lineages. And so these are the basic signatures that are going to give rise to these kinds of trees that we observe with skewed branching and multiple mergers. Okay? Yeah?
What's the rationale for having no parents having fitter offspring here?
I mean, this is just a schematic. So obviously, occasionally, parents are going to have, you're going to move around and diffuse within this fitness distribution according to, beneficial and deleterious mutations in the ancestral history. And so you can definitely move in both directions. But on average, your movement is going to be towards a more fit subset of the ancestral population. Now, of course it can be, and that's relative to the ancestral population, right? So it could be that the population as a whole is getting more fit or less fit with time or staying the same, but relative to the ancestral population distribution, you're moving to this more fit subset, and that's what's going to matter, okay? All right, so this is the basic idea, all right? And what we're going to do now to make these things a little more concrete is specialize to one specific model. And what I'm going to try to do is, at least as far as I can tell, write down the absolute simplest possible model in which we can address these questions. So what I'm going to do is imagine some population of constant size n, some overall genome-wide mutation rate u, and some distribution of fitness effects rho of s. Okay, so what that means is that any mutation that occurs has fitness cost or benefit s with probability rho of s, and some recombination rate across the genome of r. Okay, so this is a fairly simple model, and I'm going to make it even simpler. I'm going to think entirely today about purifying selection, where all these mutations are deleterious. Okay, actually, everything I say is going to apply to the case of positive selection as well, but I'm just going to focus for concreteness on the case of purifying selection so that I avoid making mistakes. And I'm going to, in fact, for most of the talk about an even simpler model, where this rho of s, it just has two components. There's some neutral mutations which occur with total rate un, and some deleterious mutations which occur with total rate ud, which all have the same fitness cost s. Okay? So this is really, I think, the simplest possible model you can write down. And I'm going to assume there's no recombination. So we're just looking at an asexual block of the genome. And then later, towards the end, I'm going to talk about how we can incorporate recombination as a sort of collection of different independent sort of asexual blocks within the genome and distributions of selection coefficients as well towards the very end. Okay? But this is really the simplest model. It only has a couple of parameters, really. N times the deleterious mutation rate and n times the selection coefficient. There are really only two parameters. These neutral mutations, of course, don't affect anything in the process. All right? So, all right, how do we understand this? So, is there some chalk somewhere here? Ah, there we go. All right. So, the approaches I'm going to talk about rely on something called the structured coalescent, which has been understood for some time. Basically, the idea is, in this very simple model, you're going to have some distribution of fitness within the population. And when selection is sufficiently strong, okay, which I'm going to talk about what happens when it's not in a minute, but when selection is sufficiently strong, we just have a fairly simple equation for the fraction of the population that has k deleterious mutations. You can write some simple differential equation to figure out what this is, which in steady state has this simple solution. Okay, the fraction of individuals that have k deleterious mutations is Poisson distributed with mean ud over s. Okay, and so you draw this distribution. Okay, and so one can imagine that we implement this process, which I talked about schematically on the last slide. You pick individuals at random from the population. They have k mutations with probability h sub k. And then we think about the ancestral history of the population. If two individuals are in two different fitness classes, they can't have coalesced. But what can happen is, as we look backwards in time, their ancestors could have less deleterious mutations, and we can think of this as a sort of migration through the fitness distribution. And given that we know HK and the mutation rates and so on, we can figure out the probability, the sort of rates of these migration events. We can follow them backward in time. If at some point in the past these two individuals have an ancestor in the same fitness class, they can coalesce, or alternatively they could continue to migrate, and eventually they coalesce. Okay, so this is just known as a structured coalescent. All right, and this allows for very efficient computation of the genealogical structure. Okay. So there's one very well-known limit. Many of you probably know this quite well, which I'm going to call the background selection limit, okay, which was introduced by Charlesworth about 20 or so years ago. And the idea is that when selection is sufficiently strong, all right, this migration through the fitness distribution happens rapidly. Okay, and intuitively this makes perfect sense. If deleterious mutations are strongly deleterious, you know, if you have any deleterious mutations, you must be very recently descended from somebody that didn't, okay? Because otherwise you would be dead. Or you would not have any, yeah, you would not be around in the first place, really, better way to put it. So anyway, so you're very recently descended from somebody in this class of individuals that had 0 deleterious mutations. And so and then within this class, right, all the individuals then are coalescing essentially neutrally. Okay, so you have these trees, these genealogies, which basically have this short delay phase, okay, where you're descended from this most fit class, or 0 class as it's sometimes called, and then a neutral coalescence within that class, okay? And so the genealogies are going to be characteristic of neutral evolution within this smaller effective population size with these tiny little branches on the end due to selection, okay? So they're more or less neutral coalescence, okay, in a smaller population with some effective size given by the size of the k equals 0 class, which if you just plug in k equals 0 here, you see that that has this population size, okay? So the selection is reducing the diversity, but not otherwise really changing anything, all right? And this becomes exact in the limit as ns goes to infinity, all right? As we take ns goes to infinity, the delay phase becomes negligible really in comparison to this. Of course, when we do that, if we don't also take nu to infinity, then we just go back to the strictly neutral result. So we also want to take nu to infinity holding this constant, okay? And then this becomes exact. And as it turns out, this approach actually works reasonably well even for finite ns, particularly if we include these corrections due to the structured coalescent where, you know, we account for the fact that these branches don't happen instantaneously, and there can be coalescence within these other classes as well. We can do this reasonably well, all right? However, you can see from this result that at some point this has to break down, right? So this effective population size depends exponentially on UD over S, and so it really doesn't take very large UD or very small s before this whole thing is just going to break down, right? Because if n effective is less than 1, okay, and in fact you don't even need that. More specifically, if ns e to the minus ud over s, okay, this is going to be our key parameter. If ns e to the minus ud over s becomes less than or of order 1, all right, it no longer makes sense to talk about this fitness distribution in this sense, okay? Because what's going to happen is these classes are going to be fluctuating very rapidly. You're going to often lose this 0 class, and when you do, it's very hard to get it back. And so you'll get Muller's ratchet clicking very rapidly. And various things are happening in this case. So first of all, we can't treat this distribution as fixed. It's fluctuating all over the place. It's no longer has this on average Poisson shape. And this NE really becomes less than 1, and that doesn't make sense either, all right? So you can, in principle, write down the structured coalescent in these fluctuating cases, and Nick and Allison have a paper from about 10 years or so ago where they work out a lot of this, but it's, at least as far as we've been able to tell, impossible to do these sort of calculations efficiently when you have these fluctuations dominating, all right? But still, sort of intuitively, something should work, right? I mean, we still are going to have variation in fitness. We can still think of these genealogies migrating through the fitness distribution. And so the question is, how do we account for these effects of fluctuations and so on, even in this situation where UD over S becomes substantial and we can no longer use this simple structured coalescent, all right? So that's going to be the goal today. All right. Yes.
In that previous slide, what's XC?
Yeah, I should have taken that off.
Sorry, it's just.
It's a characteristic. So there's various characteristic widths that come in here. So there's the standard deviation in fitness, and there's also this nose here where. Okay, that was scary. Where the population size becomes of order 1 roughly, and anyway. I'm not going to get into that. All right. So I I'm going to talk about how we analyze this. Before I do, I just want to talk about some purely phenomenological results that we get out of simulations, which are then going to motivate the approach that I'm going to take. And so, sort of inspired by some intuition, which I'll get to in a minute, what we did was we did a whole bunch of simulations across a grid of different values of ns and nu here, okay? And this line here represents the line where ns, basically ne, defined in this way, basically where this becomes of order 1, okay? And so above that line, okay, we expect this sort of structured coalescent background selection approach to work well, all right? And so that's this blue region here, all right? And below this, on the other side of this, we don't expect it to work well, all right? And that's this red region here. And these are sort of just grids of where we did the simulation, all right? And so when we do all of these simulations, this is the kind of thing that we see, okay? So here I've organized all these simulations done at a variety of different values of u and s, okay? I've arranged them according to just their value of u over s, okay? Which in this background selection, the structured coalescent regime, we expect the only thing that matters should be u over s, not u and s separately, right? From those results over on the chalkboard. And I've drawn here the simulations in that blue background selection regime as triangles, and the simulations in that red regime as circles. And what you see is that for the simulations that are denoted by triangles, okay, all of the simulations at a given u over s have the same amount of scaled diversity, okay, the same heterozygosity. Relative here, this is all scaled by the neutral heterozygosity that we would expect if there were no selection at all. So when u goes to 0, we go back to that purely neutral result. As u over s gets larger, we drop down, but simulations with different values of u over s fall on top of each other as long as they have the same u over s, and this dotted line is that effective population size result over there. But on the other hand, in this red regime, all right, you have simulations with the same value of u over s that have very different levels of diversity. Okay, so clearly nothing like this background selection approach can work here. Okay. On the other hand, there is something that works over there, which, as Richard alluded to a couple of days ago, turns out to be the variance in fitness. So if instead of organizing these simulations by u over s, I plot them as a function of n sigma, we see the opposite result, where everything in this red regime falls on the same curve. So simulations with different parameter values but the same n sigma have the same diversity. And, but of course that doesn't work for simulations in this background selection regime, where u over s is going to be the key parameter. All right? So this is just a phenomenological observation about the average levels of heterozygosity. We can also look at the frequency spectrum. It's not just that the standard deviation in fitness, the variance in fitness, predicts the average levels of heterozygosity. It also predicts everything that you would want to know about the frequency spectrum. So here's a neutral frequency spectrum here. This is just an example of a frequency spectrum in this pervasive selection regime. Okay? For these particular values of parameters, just as an example. We can summarize these deviations from neutrality via various statistics like the minor allele frequency or autogenous D, which show how sharply this falls off. And we also see this non-monotonicity here in the frequency spectrum, which is typically often taken to be a sign of positive selection, but as we see here is also present with just purifying selection, and we can quantify that as well. And what we see is that if you have a given value of pi, the pairwise heterozygosity, you have the same levels of distortion in the site frequency spectrum. And this is true for the asexual case, as well as here we've done some simulations with different levels of recombination just to show you this is not some special to the asexual case, also holds for recombination. All right, so basically, yeah.
To get it right, so this sigma would be something like a new parameter, right? Because it's the variance of the...
It's the variance in fitness, right. And as I'll discuss in a minute, so for a given value of nu and ns, one can calculate what sigma is. So this is a perfectly calculatable thing. But it's a more complicated function of u and s, okay? But it's, yeah, it's a composite parameter that turns out to be the important one here. All right?
So you infer it really from the population. So it's not going into the model that you draw a random S every time.
No.
S is fixed.
For now, S is some fixed value, U is some fixed value. Given that, that determines, yeah, exactly, as you would expect, right? Okay, so basically what this is saying, just to go back here for a moment, the standard deviation in fitness predicts pi. Pi predicts all the other aspects of the frequency distribution. So somehow, if we ought to, of the site frequency spectrum, I mean, so somehow we ought to be able to calculate everything in this regime, all right? So how are we going to go about doing this, all right? So just want to think for a minute about the sort of phase diagram of parameters here, right? We have these two parameters, ns and nu. Okay, and we have these two basic regimes which have been inspired by these simulations. Okay, there's some boundary here which, loosely speaking, is going to be this line. Okay, it's not exactly this, but loosely speaking, it's this line where ns e to the minus u over s equals 1. Okay, on this side of the line, we have this background selection regime where populations with the same lambda, which is lambda is just my notation for u over s here. Populations with the same lambda have this more or less the same diversity. This becomes exact in this limit as you go off in this direction, ns going to infinity while u over s is constant. But it still holds reasonably well throughout this regime, provided that we make structured coalescent corrections as we get close to this boundary. And on the other side of the boundary, okay, what we've seen from these simulations is that populations with different underlying parameters but that have the same value of this variance and fitness will have the same patterns of diversity. And what you can show by writing down the full stochastic version of this model is that in the limit as ns goes to 0 while holding n sigma constant, so in other words, as you go off in this direction, okay, you can show that the only parameter that can come in is this n sigma. Okay, so in this limit, kind of comparable to this limit, this sort of interference selection is actually a thing. It becomes exact, okay, where only n sigma matters, all right? And so we call this the infinitesimal limit sometimes because this limit where ns goes to 0 while holding n sigma constant is basically the classic limit in quantitative genetics, right? It's the idea where the phenotype, in this case fitness, is determined by an effectively infinite number of very small effect loci, right? So it's just the standard quantitative genetics limit that we think about. But the important thing for our point of view is sort of because of these simulations, we expect that this limit is reasonably accurate even as we sort of go down here up until we get to this boundary, right? That's what the simulations were telling us. And so what this suggests is that what we can do to calculate what we expect diversity to look like for any population over here, we take some population with some ns and some nu, we calculate the sigma, okay? And then we sort of look out, this population should have identical diversity to the one way out here in this limit where this becomes exact, and we can just calculate, all right? Now the problem is that you can't, although you can show that only n sigma matters in this limit, you can't actually at least we haven't been able to calculate anything about the diversity in this limit. I think it's probably doable, but we just haven't been able to do it. So instead we do something a little different, which is kind of funky if you think about it. So these things all have, these populations with the same n sigma on this side of the boundary should all have the same diversity. So when we take a population, say over here, what we're going to do is approximate it instead of by going off in this direction, going in this direction, and approximate it by a population right here on the boundary, okay? And this population on the boundary, this background selection, or more precisely the structured coalescent, works reasonably well. Because in fact, this boundary is a sort of fuzzy thing. There's a sort of overlap region where both approaches work reasonably well. And so what we're actually going to do is approximate the diversity in this population over here where the structured coalescent is breaking down by this population with the same n sigma right on the boundary using these strong selection structured coalescent results. So it's really a kind of a funny thing. where the weak selection becomes equivalent to a strong, so basically these weak selection with high NU and small NS becomes equivalent to a population with the same sigma, but with larger, smaller NU and larger NS. Okay, so it's a sort of equivalence between strong and weak selection in one sense of the word. All right, and intuitively what's happening here is we're essentially sort of coarse-graining the fitness distribution. When we have this sort of complicated distribution of fitness within the population, which is fluctuating all over the place, we can think of it sort of by grouping these fluctuating fitness classes together. And the kind of inspiration for this came when we were thinking about the neutral limit. So, you know, you can write down all these kinds of things, even, I mean, in principle, when S goes to 0, okay? You can still think about fitness classes, all right, even though they no longer mean fitness, right? But you can classify individuals by how many neutral mutations they have. And there'll be some distribution of that, which will, of course, be fluctuating all over the place. But not too much, right, because there's common ancestry. So there'll be some variation in fitness within the population, which will be variation in number of mutations, which will look, you know, something like this, even as S goes to 0. But the structured coalescent, of course, makes no sense whatsoever. And we get around that by just saying we're not going to keep track of how many mutations you have. We're just going to treat this as one big class, and that's the neutral coalescent. So we just thought, okay, we could do the same kind of thing, but a little less extreme as S gets small, but isn't strictly 0. And the idea is, right, here's like a left tail of this fitness distribution, just a schematic. We group mutations together, group classes together, in this sort of coarse-grained way. And the idea is, in this sort of coarse-grained framework, the effective selection pressure is larger, and the effective deleterious mutation rate becomes lower because now only a fraction of the mutations moves you between classes, right? And so that's essentially what we're doing when we're going in this direction, all right? And of course you have to do this correctly, right? You can't coarse-grain too much because otherwise you're neglecting important differences in fitness, and you can't coarse-grain too little because otherwise the structured coalescent won't work. And it's, you know, not at all obvious there is a coarse-graining that will work, but in fact there is, and these sort of simulations tell us or inspire us, and this infinitesimal limit inspire us to suggest that the correct coarse-graining is the one that preserves n sigma, okay, while going all the way down to this boundary where this becomes of order 1. Okay? And so, all right, how well does it, so first of all, okay, obviously this creates an issue of identifiability, right? It's saying that any population that lies on this line, that lies over here, is going to look like a population on this boundary. This boundary is the place where NES equals 1. So, you know, whenever you infer NES equals to 1 in these kind of contexts, it's also, everything is also consistent with these other parameter values here. But the advantage of this is it allows us to calculate. So for example, if we look at a slice, this is looking at a slice across here, right? Initially we get a reduction in diversity as we go towards higher and higher U over S. And then we cross this boundary and we're going on this side now. And what's happening is as we go on this side, we're mapping down in these directions sort of effectively along this line, and the diversity is coming up again until we eventually regain the neutral limit on the other side as S goes to 0, as we had better do, right? And more generally, right, you can see this red line, which I didn't talk about before, is the prediction of this coarse-grained model, basically calculating n sigma given U over S, U and S, and predicting, and you can see it works really well. And it also works not just for the overall levels of diversity, but also for the full site frequency spectrum. So here are a bunch of different site frequency spectra for different underlying values of the parameters, which I'm hiding for now, but which I'll tell you in a few minutes. And this is the prediction for that value of n sigma for the site frequency spectrum. So it's not perfect, obviously, there's some issues here, but on the whole, it predicts things reasonably well. And to come back to something that Richard was talking about a couple of days ago, right? So this is the neutral expectation up here. And this is the Bolthausen-Snitman coalescent prediction down here. All right? And this obtains basically in the limit as n sigma goes to infinity, right? So n sigma going to infinity corresponds to the Bolthausen-Snitman. But of course, there's a whole lot of room in between, and with this coarse graining, what we can do is predict all of these things in between. So Richard showed the influenza data, or actually just some simulations, right, sort of spanning this full range. And what this allows us to do is sort of interpolate properly between this which basically is n sigma equals to 0, and this which is n sigma equals to infinity. Okay? Now, right, and yeah, it just predicts all of these distortions across. That was just one example. It sort of predicts across all these regimes reasonably well. All right? So now I want to talk about two complications to get back to the original model that we started with, the simple one, not the simpler one, right? So the first of these is thinking about recombination. And I'm not really going to get into this in detail because Richard basically outlined the idea a couple days ago. The idea is basically, just to reiterate what Richard said, you have some genome, very distant sites that recombine many times in the time to the common ancestor are basically freely recombining, and we can treat them as independent. On short distance scales within the genome where recombination doesn't typically occur within the time scale of the most recent common ancestor, we expect things to be effectively asexual. And so basically we expect to be able to treat the genome as a sort of collection of these linkage blocks, okay, where the size of the linkage block is basically, this is the scale on which recombination doesn't act in the time to the most recent common ancestor. And as Richard mentioned, of course, the deleterious mutation rate within the block, okay, depends on the length of the block. That deleterious mutation rate, of course, affects this time to the most recent common ancestor. And so these things are not independent, and one has to solve for this self-consistently. But in the things that I've shown you, basically, we can do that self-consistent solution. And so you can see, for instance, for a given value of U and S as a function of the recombination rate, we can predict the diversity. These blue things, I guess, yeah, I don't want to talk about them. I don't really have time. But this is a sort of prediction of our approach implementing this sort of crude linkage block approximation. I think there's a lot of work to be done to make this better. I mean, this is really just ad hoc and totally, unjustified, but it appears to work reasonably well. And so we want to think about that a bit more. The one other thing I want to talk about is when we think about distributions of fitness effects. So this is also something that we're working on now but really haven't solved. Imagine you have some distribution of fitness effects where there's some weak effect mutations and there's some strong effect mutations. Intuitively, what we expect to be able to do is take all the weakly selected mutations, okay? And these things, it really doesn't matter what the distribution is. Basically, all that matters is how these weakly selected mutations determine the variance in fitness, okay? And so we can look at that just based on some effective selection coefficient and how it determines the variance in fitness. And then these strongly selected mutations basically live in this regime, right? And they're just reducing the effective population size, okay? And so somehow if we can couple these two things properly, we ought to be able to predict things. And of course, we have to have some regime in the middle, which is the boundary between these two things. And where this boundary is depends on things like the mutation rate. If we increase the mutation rate, this boundary is going to shift in this direction. Or if we decrease it, it's going to shift in this direction. So this is going to lead to all kinds of complicated scaling of the diversity with the actual population size and mutation rate. And so we can now solve this in certain simple cases. Like if all the mutations are relatively weak, we can solve this. If there are just a few different classes of mutations, we can solve this. But in the general case, we're still trying to figure out how to do this. But just to show you that in principle this should work, I'm going to now tell you what these different things are. So these different site frequency spectra, which I'm showing you, are for a variety of different populations. In this case, one that has some non-zero recombination rate, different sorts of distributions of fitness effects, a single S model, a finite sites model. And what you can see is in all of these cases, right, these cases are chosen so they all have the same variance in fitness. And given that they have the same variance in fitness, they seem to have basically the same site frequency spectrum, and it's predicted by this model, even though the underlying details are very, very different. All right? So I think I'm about out of time, so I'm going to stop there. And just want to thank a few people in particular. So this is my group. And this work was all done by a really fantastically good graduate student, Ben Good. And as I said before, it's also in collaboration with Alex and with Richard in particular.